Very good morning, dear students. We have been discussing the details about enzyme activity. What are the characteristic features of an enzymes and how enzymes catalyzes the biological reactions, and what are the major factors which influences the enzyme catalyzed process, such as the substrate concentration and the temperature, pH, etc. we have discussed. Then we have also discussed the Michaelis-Menten concept and Lineweaver-Burke equations and their mechanism of enzyme activity we have discussed in the previous class. So once the enzyme activity is prolonged, so once the process stops, it should be adequately controlled and it should be regulated actually. And this particular step is very important in all the enzymatic process. Once the enzymatic reaction completed and the product is produced, the process has to be stopped and it should be terminated. Okay. And in prokaryotes and eukaryotes, there are several mechanisms for the activity of the regulation of the enzyme catalyzed process. Okay, and that is what the next important topic. So the next topic is regulation of enzyme catalyzed reactions. So first of all, we will discuss what are regulatory enzymes and what is their necessities. And a regulatory enzymes is the enzyme that is commonly present in a biochemical pathway, which through its response to the presence of certain other macromolecule or biological molecules, which regulate the pathway and terminate the process. So a regulatory enzyme is the enzyme in a biochemical pathway which through its response, once it is responded, what will happen? The presence of certain biomolecule in the pathway will be getting regulated. So that is known as regulatory enzyme. So this is usually performed for the pathways whose products may be needed in different amount at different times, such as hormone production. Okay, so always it is not required, and the cell required this particular component at certain times at a particular concentration. Okay, and that particular concentration, once it is produced, and once the cell require adequate quantity of this particular enzymatic activity, when the product is produced, this will be properly regulated. The regulatory enzymes exist at a high concentration, that means to say lower V maximum, and their activity can be increased or decreased with the changes in substrate concentration. Okay, so the the regulatory enzymes generally exist at a high concentration, so that their activity can be increased or decreased with the change in the substrate concentration. That is what I told you. Substrate concentration is very important. That is what the Michaelis-Menten concept dealing with actually. So the enzyme which catalyze the chemical reaction again and again are called regulatory enzymes. So the enzyme which catalyze the chemical reaction repeatedly is known as a regulatory enzymes. So what are the various mechanisms for enzyme regulation? The main enzymatic activity and the regulation of enzymatic activity or control of enzymatic activity is achieved by mainly three mechanisms. Okay, number one, zymogen activation. Number two, covalent modification. Number three, feedback inhibition. So the major regulatory mechanism of enzyme activity are zymogen activation, covalent modification, and feedback inhibition. Then we will discuss all this concept one by one in details. So what are zymogen activation? So what are zymogens first of all? Some enzymes, especially the digestive and coagulating enzymes, are synthesized in the body in its inactive form. When the enzymes synthesize, you know that is protein. That proteins, once it is synthesized, okay, so they will not be in their active form. Okay, so this inactive form of enzymes are known as zymogen or proenzymes. Okay, and these are the precursors of an enzyme. The previous inactive form of an enzyme before it is getting converted into an active form is known as the precursors. Okay, so they are not active on their own but can be activated after the conversion from inactive form to active form. Okay, then how will you represent this zymogen? The zymogen can be represented either adding the prefix pro or adding ogen suffix. Okay, or adding the suffix ogen is added to the enzyme's name to denote its zymogen, such as prothrombin, proelastase, trypsinogen, pepsinogen and fibrinogen. Okay, so here prothrombin, there is a pro, the thrombin is an active form, prothrombin is an active form. Elastase is an active enzyme, proelastase is an inactive enzyme. Then here is again pro. Then I told you sometimes can be ogen, that is trypsinogen is an inactive form, trypsin is an active form. Pepsin is an active form but pepsinogen is an active precursor. Fibrin is an active form and fibrinogen is an inactive form. That is what I told. Either pro can be added or ogen can be added to denote the precursors or the inactive form. And this precursor should be converted to the active form before they can catalyze the chemical reaction. And this type of activation usually involves the cleavage of the peptide bond. Enzyme, you know that long chain polypeptide. When the enzyme is getting synthesized, I told you they are disulfide linkages, and these disulfide linkages will be giving the stability, and that has a lot of importance in the denaturation and the stability of the enzyme. So how this zymogen will be activated? We will take one typical example how chymotrypsin will be converted into trypsin. Okay, so chymotrypsin is synthesized by exocrine cells of pancreas. Pancreatic exocrine cells that is present in the form of a precursor that is known as chymotrypsinogen. Okay, chymotrypsin is synthesized in the body in the form of chymotrypsinogen. The hydrolysis by the enzyme called trypsin convert chymotrypsinogen into an active form called chymotrypsin. So what are the basic molecular biology mechanisms are involved here is the chymotrypsinogen is composed of a single polypeptide chain with a 245 amino acid in length and 5 intra-chain disulfide bond. That is, I told you that has 5 different types of intra within the polypeptide chain. There are 5 different intra-disulfide bond. So it is lack of enzymatic activity and it's converted into a fully active enzyme when the peptide bond linking arginine 15 and isoleucine 16 is cleaved by trypsin. Okay, the first there is a cleavage is happening between arginine 15 and isoleucine 16 by trypsin. Then that resulting component called pi chymotrypsin, that is an activated one, and again this pi chymotrypsin molecule that is activated by 2 disulfide bond. Okay, that is between 14 and 15 and 147 and 148 and removed to yield an alpha chymotrypsin. So pi chymotrypsin will be released to form alpha chymotrypsin. This is a stable form of active enzyme. So this resulting chain, it has to go to 3 chain, A chain, B chain and C chain in alpha chymotrypsin, which are covalently linked to each other by 2 interchain disulfide bond. Okay, so this cleavage of the single specific peptide bond transform the protein from the catalytically inactive form into one that is fully active chymotrypsin. So this is what I have told, dear students. So here it is a 255 molecule chymotrypsinogen, that is in inactive form. It has got 5 different disulfide linkage between 14 to and 58 number 1, 168 to 182 number 2, 191 to 220 number 3, and 1 to 122 that is number 4, 136 to 201 number 5. Okay, so then what will happen? There is another enzyme called trypsin. This trypsin will go and bind with this particular trimotrypsinogen, and they will cleave between arginine and isoleucine 15 and 16, and this fragment is gone. Okay, and there is a breakage, and that is known as pi-chymotrypsinogen. That is an active form. Then again what will happen? This pi-chymotrypsinogen again will be undergoing some small rearrangement cleavage. That is two dipeptide, that is serine 14 and arginine 15. Serine 14 and arginine 15 will be cleaved, and tyrosine 147 and asparagine 148 will be, threonine 147 and asparagine 148 will be cleaved. Okay, then you will be having 3 fragment. Okay, 1 to 13 1 fragment, then 14 and 15 is gone. Okay, 16 to 146 is another fragment, then 147 and 148 is also gone, then 149 to 245 another one. Okay, 1 2 3, this is what the A chain, B chain and C chain. Okay, this is fully active. That is, these 3 functional unit are known as the chymotrypsinogen. That is known as alpha chymotrypsin. This is a active form. Then similarly, the trypsinogen can be used in pepsin, and that can be converted into trypsin. Okay, trypsinogen is an inactive form, peptin is an enzyme, and that converted into trypsinogen. Similar type of mechanism is happening. I am not going to discuss all those details here. Similarly, pepsinogen, an enzyme called enterokinase, so it produces pepsin. This is inactivated, fully inactivated, and this is fully activated, plus a polypeptide will be produced. So this is known as the activation of zymogen. So activation of zymogen will be happening after the cleavage of an enzyme using cleavage an enzyme in the inactive form of an enzyme that convert into the precursor, that is an inactive form into an active form. So that is the first mechanism. The second one is covalent modification. The activity of serine-containing enzyme. There are so many serine-containing enzymes. We have already discussed the classification of an enzyme. Serine, you know that one of the important amino acid. So that there are so many serine-containing enzymes which is playing a very vital role in the regulation. And how it will be? Mainly a covalent modification, the covalent insertion of a small group on the enzyme molecule. So serine-containing enzyme is regulated by inserting a small group to that particular enzyme. So what is this particular group actually? For example, the activity of the enzyme that synthesizes the cleavage, the glycogen, is regulated by the attachment of a phosphoryl group to the specific serine residue. Okay, the attachment of this small molecule can be a small phosphate group. Okay, so once the phosphate group is added, it will be modified and the enzyme will be regulated. The active site of the enzyme is changed and it cannot be accommodated the substrate, and it has got regulated. So this modification can be reversed by hydrolysis. Once it is going hydrolysis, this will be gone and the enzyme again can be continued. Okay, that is known as covalent modification. So serine-containing enzyme mainly doing this particular process by adding certain small molecules such as phosphate group to the binding site or the active site of the enzyme, that will be modified and the substrate cannot go and bind to the active site and the enzymatic activity cannot be continued. So this is the next important mechanism. The third one is feedback inhibition. Okay, so this is very common in the synthesis of some amino acid. Okay, so some enzyme catalyzes the synthesis of small molecules such as amino acid in number of steps. Okay, the amino acid synthesis from the starting compound, it includes a lot of steps. Okay, the enzyme catalyzing the first step in this biosynthesis is inhibited by the end product of the reaction. This regulatory mechanism is known as feedback inhibition. Okay, the enzyme catalyzing process. Okay, the enzymatic reaction from the from first component, it will take a lot of steps and it will be producing an end product. That end product will go and bind it to the enzyme that is acting on the first step of the reaction. Then it will be completely inhibited, it will not be done. Okay, that is known as a feedback inhibition. So for example, the biosynthesis of isoleucine from threonine. Okay, so the biosynthesis of isoleucine from threonine. So once the isoleucine is produced, what will happen? This will go and bind with the threonine. The first step of threonine the conversion of threonine, it will be inhibited the synthesis of isoleucine. So this is what I have told. So here you can see the component A, B, C, D and E. Okay, there you can see the component A is the starting compound. Then once after some enzyme, activity of some enzyme, it converted into B, and the B using some enzyme, it converted into C. C catalyzed by some enzyme and it produced D, and finally D, the enzymatic activity by some enzymes, and it is converted into E. Okay, once E produced, this end product will be acting as the inhibitory substance. That is known as they will go and bind to the first step and they will block this conversion of A and B. So the entire pathway will be getting blocked. So that is known as feedback. Okay, this is feedback inhibition. This is end product will be inhibited at the initial step of the enzymatic activity. So this is all about a brief up about enzymatic activity. So dear students, today we can conclude what are the major important points we have discussed here is initially we have discussed the regulatory enzymes. Okay, introduction to regulatory enzyme. What are regulatory enzymes? What are the major enzymatic activity of regulatory enzyme? The major mechanism that has got 3 important steps: cymogen activation, covalent modification, feedback inhibition. Cymogen activation. What are cymogen? How it will be getting activated by taking an example of a chymotrypsin to trypsinogen, trypsinogen from chymotrypsin. Covalent modification, okay, how it is modified, especially serine-containing enzymes, okay, how it is getting modified by adding the phosphate. Then the feedback inhibition, the synthesis of threonine from isoleucine is excellent example of feedback inhibition. That's all, dear students. So these are the textbook Principles of Biochemistry by Leninger and Biochemistry by J.L. Jain we can refer. So all those textbook also you can refer. So thank you, dear students, for watching this particular class. If you are having any clarification, you can write it to me, or you can post here. Thank you very much.